Hi, this is Denise here with healinghacker.com, and today we are talking with Associate Professor Christopher Lowry, and he works in the Department of Integrative Physiology and the Center for Neuroscience at the University of Colorado in Boulder. And thank you so much for talking with me today, Professor Lowry.
You're welcome. It's nice to be here.
So let's jump in. Can you tell us a little bit about your area of expertise and study? And how that leads us to, we're also going to talk about the hygiene hypothesis, kind of bridge that and fill in the spaces for us.
Great. So I'm a behavioral neuroscientist, so that means that I study mechanisms that control behavior. Our focus is emotional behavior, so behaviors that are involved in anxiety and affective disorders like anxiety, fear, affective behaviors, also cognitive behaviors. Historically, the focus of our lab was on the brain. More recently, we've become interested in questions about how the body communicates with the brain, and then how those signals are controlling emotional behaviors. Part of that interest arose around the year 2000, when we became interested in studying a bacterium called Microbacterium bacchae. And our original experiments were designed to understand how these antigens, or antigens from these bacteria in the lungs, could affect brain function and behavior. And in addition, we have a very strong focus on serotonin in the brain. So how do these signals affect serotonin in the brain, which is a neurotransmitter? And then even more recently, that's expanded to try and understand how not just one bacterium, but communities of bacteria, like what you find in the gut, can affect the brain and behavior.
Okay. So that kind of leads us to the popular term is the hygiene hypothesis, and I noticed in some of your writing that you call it the old friends mechanism. Why the distinction? Can you talk about that, and also talk about that in light of another term that you talk about, which is old infections?
Right. So the old friends terminology was put forward by my colleague and friend Graham Rook at University College London. He's an immunologist, and he and his colleagues, I think, wanted to distinguish between the original hygiene hypothesis and our current understanding of the mechanisms that are involved in the hygiene hypothesis. So the original hygiene hypothesis was based on the idea that, or observation that, children that had older siblings had protection from inflammatory disease. And it was thought that that's because the older children are causing more infections than the younger children, and it's the infections that are providing all this protection. It's become clear now that it's not really the infections, like getting a bacterial infection or a viral infection, that's protective per se, but it's exposure to certain types of microorganisms, many of which are not pathogenic at all, but come from our environment. And now the idea is that if you have older siblings, you're exposed to more environmental antigens or environmental bacteria, and that provides protection. So it's simply a way of thinking about it differently. And these old friends have several categories. One of them is the old infections that you mentioned. And so the old friends encompass basically three different categories of organisms. One is commensal bacteria that can actually colonize the body. So these include bacteria that can actually colonize the mucus layer lining the gastrointestinal tract. That includes bacteria like lactobacilli and other bacteria that actually live in the mucus layer and colonize the host. It also includes what are called the old infections, and these were basically infections that were transmitted by the oral fecal-oral route very early in life, so in children. And what's common among these old infections is that they were highly ubiquitous. So most people had them. But on the other hand, they're infections, but they're not going to kill the host. And so that's what allows these types of infections to be so prevalent throughout human history.
What kind of infections would those be?
So these include some types of hepatitis virus, toxoplasma, and Helicobacter pylori is a good example. I like Helicobacter pylori as an example of an old infection because there's quite a lot known about it. Martin Blazer has written a book about this, which I highly recommend. Essentially, this is a bacterium that people don't really know what to call it because it's commensal, and throughout human evolution, it was virtually ubiquitous in all humans. And this was at least for 66,000 years, maybe over 100,000 years, we've co-evolved with these Helicobacter species. Other people have called them not commensals, but something called pathobionts. And this is kind of a mixture between a pathogen and a symbiotic organism. And the point is that Helicobacter is normally present, but under some conditions, which are not entirely understood, these Helicobacter species expand and they can become pathogenic and cause disease like duodenal ulcers, for example. Which is why starting several decades ago, the medical profession launched initiatives to try and eradicate Helicobacter species because of this pathogenic potential.
Yeah.
But it's an old friend for a totally different reason. So during early development, these old friends bind to immune cells in the body, and they activate mechanisms that induce immunoregulation, which is another term for immune tolerance. And some of the pathways involved in this induction of immune tolerance by Helicobacter, for example, have been worked out. And early in life, when these infections were normally very common, what's happening is the presence of these bacteria changed the function of dendritic cells, which are cells that can bind bacterial antigens. And then these dendritic cells are training the immune system to be tolerant. And without that training, you have an immune system developing that lacks immune tolerance. And then you have the potential for the immune system to attack things it shouldn't be attacking, like yourself, like your gut microbiota, like bacteria that should normally be tolerated, but under those conditions are not. Yeah. And so that's what Helicobacter is doing early on in life among all the other old friends. So that's a good example of an old infection. And then the last category is environmental bacteria. And these include environmental saprophytes, which are bacteria that live in decaying organic matter. And they don't colonize the human body like Helicobacter pylori does. But throughout human evolution, they would have been passing through the human body in large quantities because they're in the water and they're in the mud and on the food. And of course, in the last 50 years, with purified water sources and more hygienic food, perhaps in some people consuming less fermented food products, there's a lack of exposure to these environmental organisms. Mycobacterium vaccae is one of these environmental saprophytes. So it's in the water, it's in the mud, it's in the soil. Throughout human evolution, we would have been exposed to large quantities. The particular strain that we're studying was isolated in around Lake Kyoga in Uganda. So it's kind of at the center of human evolution.
You know, just because you started talking about the H. pylori, which I've read Dr. Blazer's book, and it is amazing. You know, you talk about also that like 10% of our bodies are made up of our cells, and the rest of us is, you called them, I think, symbiants. What does all this mean, given it seems like we're on this gradual campaign to obliterate all germs, like H. pylori's been virtually destroyed in the US. All germs and all things gross, we're trying to get rid of it, and 90% of our bodies are made up of that matter. What does that mean?
So it means we might have to think about the self in a different way, because the self is not just human cells, but it's all of these bacterial cells. And I think we used to think of these bacterial cells, and like you said, they far outnumber our human cells. These bacterial cells are not just passive bystanders. They're actually producing chemicals that are modulating our own physiology. And they have the capacity to interact with our immune system in ways that can control both our physiology and our health, our medical health, but also our mental health. And so we're only now starting to understand some of the mechanisms through which these bacteria are communicating, not just with the superficial layers of the human body, the mucosal lining of the gastrointestinal tract, for example, but also through various mechanisms communicating with the brain and affecting behavior. And so when we think about these bacteria, we really have to think at ecological level or a community level. These bacteria, this bacterial community is extraordinarily complex. Now, that's not to say that there aren't, I think Martin Blaser uses the term keystone species. It's like the wolf is a keystone species in its ecosystem. There may be bacteria that are keystone species and have a really dominant effect on both the bacterial community and the host. But those distinctions we don't really have a good understanding of yet. I mean, are there keystone species in the human gut microbiota? And should we be paying particular attention to those species? Or should we just be looking at the big picture? In terms of the gut microbiota, one clear property of the gut microbiota that is a biomarker of good health is the diversity. And that may sound kind of vague, but you can think of it in very simple terms as the number of different species that are present. And you might think, well, why would that even matter? Why would more species or more diversity of species be better? And we don't have all the answers to that, but there are some clear ways to think about this. One is that if you have a diverse ecosystem of bacteria in your body, then it's harder for pathogens that are known disease-causing agents to get a foothold and expand and cause their disease pathology. That's been demonstrated experimentally in animal models. So in other words, stress is one of the common consequences of stress based on animal studies is a decrease in the diversity of your gut microbiota. And when that diversity declines, pathogens that cause pathological and physiological changes are able to expand in those models. And so there's some empirical evidence that diversity is good, lack of diversity is bad. The problem is that, or the question then becomes, how do we maintain a highly diverse gut microbiota in order to have more resilience to pathogens and stressors?
And also, it's interesting because how do we maintain that? I feel like there's a thrust in this, in our culture here, to, everything should be antibacterial, everything should feel nice and clean and almost sterile, for lack of a better word. And we don't even know what we're killing off because now it's gone. And how do we get it back? So it's,
for people that are interested in those questions, Martin Blaser's book is a very good book. Jessica Sachs also has a very good book called Good Germs, Bad Germs. It was published a few years ago. These really get at that question. You know,
Yeah.
what are we doing really when
Yeah.
we're pursuing these goals of greater hygiene and less exposure to germs or bacteria.
Yeah.
And I think what's becoming clear is there's a price. Sure, it's very clear that we're, as a population, we're much less at risk from dying from infectious disease, which of course is a very good thing. And much of that success has been through the use of antibiotics. But there's a cost to that,
Yeah.
that benefit. And we're only now starting to learn something about the cost. One thing to point out is that most antibiotics, when we think about antibiotics that we take, where we get prescriptions from the doctor, we take these antibiotics. Most antibiotics were derived from bacteria. And so this is something people don't often think about. But bacteria are effectively at war with each other. And bacteria make antibiotics specifically to prevent the proliferation of competitors that are competing for resources that they need to survive. And so most of the antibiotics that we use are compounds that were isolated from bacteria or other microorganisms that have this antibacterial property. And a vast majority of these antibiotics come from a phylum of bacteria called the actinobacteria. And the actinobacteria are interesting because these are the environmental bacteria. These are the ones that, well, they're not all environmental, but many of them are environmental, and they include the mycobacteria like Mycobacterium vaccae.
That is interesting. I did not know that. I want to talk a little bit about, so the whole idea, in one of your studies I read that you had germ-free mice, and I know this is a mouse model, and there's lots of stuff that we won't talk about here, mouse model versus human model, but you had sterile mice that had aberrant behavior, abnormal nervous system structure, all these abnormalities going on, and then and maybe I misunderstood it, so correct me if I'm wrong, you repopulated their gut microbiota before 6 weeks, and you were able to kind of reverse all of those abnormalities. If you could talk about that a little bit, but also talk about what that could potentially mean for humans as far as like correcting existing issues, what the theoretical windows would be, and what that might look like.
Okay, so just to quickly clarify, that's not our work, but we've reviewed that work and
Yes.
discussed that work. And effectively, there's a critical window during early development, and in mice, it's the first 6 weeks of life. And in humans, perhaps a late second trimester, third trimester, extending out to 2 years of life. And the way we think about this in our lab at the moment is that critical window corresponds with when your immune system is developing the capacity for immune tolerance. And so, the capacity to tolerate pollen, dust mites, other environmental allergens, to tolerate self in the context of autoimmune disease, and to tolerate perhaps the gut microbiota. And some of the mechanisms are known, but there's a particular type of immune cell called the regulatory T cell. And production of these regulatory T cells in significant numbers during that developmental window requires exposure to these microorganisms that promote immunoregulation, like Helicobacter pylori, for example. Without that training, if you will, you'll you would have a reduced capacity for immunoregulation as an adult. And the reason is because that's when the T regulatory cells are being born as a cell type. That's the developmental window for the birth of T regulatory cells and seeding of the tissues. And so that at that time, decisions are being made whether to make T regulatory cells or something called T effector cells that can drive inflammation. And so it's a balance. It's a balance between the anti-inflammatory regulatory T cells and the pro-inflammatory Th1 and Th17 type T cells that can drive inflammation. And so we think that's why that window of time is so critical, because this balance between anti-inflammatory mechanisms and pro-inflammatory mechanisms is the stage is being set at that time. Now, it doesn't mean that as an adult, it doesn't mean that you can't modify that balance. You can, but your capacity to modify that balance is reduced, right? Because you've missed the window for the birth, controlling the birth and seeding of all your tissues with these regulatory T cells. It's clear that with the right stimuli during adulthood, you can promote or activate or even induce a proliferation of these immunoregulatory cells. And so it's not that you can't shift the balance as an adult, but the capacity to shift the balance during development is much greater.
Got it. Yeah. It just seems like you probably have so much more ground to cover than you would have in the beginning.
Yeah, the stage has been set, so to speak, and you have all of these T cells populating your tissues, and that's a lot of T cells. And so that's what you start with, and then you can modify them, right? But during development, you're actually determining that ratio. The exposure to these microorganisms is determining whether to bias toward immune tolerance or to bias toward immune activation. Now, you can imagine in evolutionary terms why that's adaptive. For a human in the third trimester, the environment during the third trimester in terms, so one of the things that's driving whether you make immunoregulatory cells or pro-inflammatory cells is the immune molecule environment, right? I want to use the word cytokine, but I haven't defined that. But cytokines are molecules that are released from immune cells, and those molecules, whether they're anti-inflammatory molecules or pro-inflammatory molecules, are helping make the decision about what type of T cells to produce during the third trimester and first 2 years of life. And you can imagine that if the mother's environment is a very pathogenic environment characterized by lots of inflammation, it would be adaptive for the offspring to anticipate an environment that has lots of pathogens. And then it may be adaptive to bias toward a more pro-inflammatory immune system. But in the absence of that inflammation comes with risks. So in the absence of that inflammatory environment, it would be more adaptive to not take the risk of autoimmunity and allergies, etc., and bias toward immunoregulation, because the risk of infection and other pathogens, the consequences of pathogens is lower.
Well, let me ask you this. You talk about, or in the piece that I read of yours that you wrote, so this might not be your term, but farming the microbiota. And so I understand, I've actually heard before that 2-year window, but you explained it really well in a way that I hadn't thought about it previously. But what actions beyond the 2-year window how does farming the microbiota, how would that look? How can that be something that could make up ground after that first 2-year window if someone's missed it and they're an adult or they're an adolescent?
Yeah, so farming the microbiota is a nice term, and effectively we're just saying that you're intentionally modifying the microbiota in a way that's beneficial. And it's analogous to farming because bacteria have food nutritional requirements just like we do. And there are certain aspects of our diet that can promote beneficial bacteria. And so an example of that is something called prebiotics. And prebiotics are indigestible fibers from plants, typically, that we don't have the enzymatic capacity to digest, but certain types of bacteria do. An example is bifidobacteria. And these are, members of the Actinobacterium phylum. And these are good bacteria. And so by consuming more indigestible fiber, you're feeding bifidobacteria. And one theory is that these, along with lactobacilli, are altering the pH in the gut, which makes it less favorable for the bad bacteria. And so there, you're basically making decisions to consume more prebiotics or indigestible fibers that will promote the good bacteria and ideally will make it less likely that bad bacteria can proliferate and get a foothold.
Well, let me ask you this too. Is it conceivable to, since you're saying that 10% of our cells are human cells and 90% are symbionts, is it possible to farm that 90% to make up for where that 10% is lacking, since it seems that we've outsourced so much of our metabolism, our immune function, so many things we've outsourced already to that 90%? Is it conceivable that, you know, to make up more ground, like you were saying, and generate more T regulatory cells, can we farm that 90% in a way that helps make up for where the 10% is deficient?
I think that is a really attractive approach. We don't have the empirical evidence for, exactly how to do the farming, but I think the farming is a really good idea, and this is why I think that's a good idea. If you think about inflammatory disease, which is basically this scourge of modern urban societies, it's one of our biggest health threats. There's various ways to approach limiting inflammation. And if I go in sequence, I'll kind of walk you through the different layers of that onion. One would be to do what we just said, is use prebiotics to promote the proliferation of the good bacteria that can compete with the bad bacteria. So that's a prebiotic approach. Another approach is probiotics. And in terms of probiotics, in that case, you're not feeding a whole genus of bacteria or multiple genera of bacteria at a community level. You're adding 1 or 2 or 10 or more different species and strains that ideally have been proven clinically to have health benefits. And so there you're tipping the balance toward bacteria that are known to have health benefits, and hopefully there's some downstream consequence for them to outcompete the bad bacteria. And some of those probiotics are known to have immunoregulatory properties. In other words, they can promote the proliferation of these T regulatory cells, which will then shift the balance from inflammation toward anti-inflammatory processes. And so we're still learning about which types of bacteria have these immunoregulatory capacities, but it's clear that some of them do. Those are the ones that we're interested in clinically. in terms of moving forward with clinical trials. A third layer, so prebiotics, probiotics, third layer is immunoregulation itself. And so you don't have to use prebiotics or probiotics to induce immunoregulation. You can do that by injecting immunoregulation inducing agents into the body and directly interacting with the immune system. So most of our research is using that approach, where we're not putting things into the gut as a nutritional supplement. We're actually injecting bacteria into the body. That trains the immune system to be more tolerant, so there's less inflammation, more anti-inflammatory responses.
Can you talk about that a little bit in detail? Because I know that, and we've talked a little bit about MVACI, which I think you've studied as well. Can you just talk a little bit about injecting bacteria and how that modulates the immune system?
Sure. Let me come back to that after. Let me just mention these other two levels, and then I'll come back to that. The other two levels beyond immunoregulation are anti-inflammatory drugs, and this has been the historical approach of medicine, right? Is to say, okay, TNF alpha is a pro-inflammatory cytokine. We know that's driving inflammation, and it's part of the disease process. So let's treat with an antibody that can block TNF alpha. There are several of those that are currently on the market. And that's not a bad strategy. It's just not a comprehensive strategy, because inflammation is not just TNF alpha. There are many pro-inflammatory cytokines, and not all inflammation-promoting molecules are cytokines. So histamine is involved in the inflammation. Prostaglandins are involved in inflammation. So if you every time you take a step back or upstream in the process, you have the opportunity to control a wider swath of the inflammatory process. And with an anti-inflammatory drug targeting a single pro-inflammatory molecule, yes, it can be effective, but it doesn't have the potential to affect inflammation on such a broad level as the upstream levels. And then the final level is simply avoiding things that through lifestyle choices that can promote inflammation. This includes things in our diet, the amount of sleep that we get, and the amount of exercise that we get. So going back to the immunoregulation, that's really where we've put all of our effort. And this is based on the belief that if you can promote immunoregulation directly, then you could have a pro-inflammatory gut microbiota, or a gut microbiota with a pro-inflammatory potential, but the immune system won't respond because it's tolerant. It tolerates those potentially inflammatory agents like the gut microbiota, like pollen, like dust mites. And so the approach there is simply to directly train the immune system to be more tolerant. We don't have to be attacking pollen or dust mites or certain types of gut microbiota. And the way we're doing that is by injecting this bacterium that I mentioned earlier, which is Mycobacterium vaccae. And interest in this particular bacterium arose many years ago, not from my lab, but through Graham Rook and his colleagues. And they're immunologists, and they noticed that the efficacy of tuberculosis vaccines throughout the world varied depending on the geography. And so there were some geographical areas where the vaccines were incredibly successful. So they went to one of these areas around Lake Kyoga in Uganda to try and understand why. And it turned out that the shores of the lake were lined with this kind of orange slime. And this orange slime turned out to be Mycobacterium vaccae. And through studying the immunology, they realized that this is a very potent immunoregulation-inducing organism. In terms of tuberculosis, it was acting as an adjuvant because it's very closely related to Mycobacterium tuberculosis. But then they realized it has this immunoregulation-inducing property. And it was found in animal models that you could give this Mycobacterium vaccae either by injecting it weeks before an allergen challenge in mice and attenuate the allergy response. And they also showed that you could give it orally, so give it into the stomach, and it could also promote this attenuation of allergy in animal models. And that immunoregulation is thought to be dependent on activation of these T regulatory cells, but it's known to be dependent on the anti-inflammatory cytokines like interleukin-10 and TGF-beta, which I haven't mentioned before. But if you're talking about inflammation, those are the good cytokines. They're the anti-inflammatory cytokines that can keep inflammation at a reasonable level. So that's really where we're focused now, is trying to understand if we can enhance immunoregulation and suppress inflammation, can we see health benefits? And of course, the previous work was all done with allergy models. My lab is interested in psychiatric disorders. So you might think, well, what does this have to do with psychiatric disorders? But it's becoming more and more clear that many anxiety and affective disorders, like post-traumatic stress disorder, depression, bipolar depression, are associated with exaggerated inflammation. And there's some evidence that if you have exaggerated inflammation prior to a stressor, prior to trauma, then that increases your risk for going on to develop PTSD. It's also very clear that if you just look at PTSD, people that have, let's say, been exposed to trauma, some have PTSD and some don't. In fact, the vast majority don't have PTSD, but a minority do. If you compare those groups, it's clear that the ones that go on to develop PTSD have exaggerated inflammation. The same is true with depression. So a study at Whitehall showed that, and this was over 3,000 people, it was a huge study. They showed if you just measure one of these, two of these pro-inflammatory molecules, interleukin-6 and CRP, or C-reactive protein, and then wait 11 years and then do psychiatric measurements, it turns out that people that had elevated interleukin-6 and C-reactive protein, these biomarkers of inflammation, the higher, the people that had higher levels of those molecules had a greater risk for depressive symptoms 11 years later. There's a more recent study that was done in Bristol in the United Kingdom, which is where I worked for 12 years. It's called the Avon Longitudinal Study, and it's a study of children and developing children. And they measured interleukin-6, I think it was in 9-year-old children. And then they looked at depressive symptoms and psychotic symptoms at age 18. And again, it was the children that had elevated interleukin-6 at age 9 that went on to develop depressive and psychotic symptoms at age 18. Wow. And so it looks like a risk factor. Now, obviously, we have to point out this is just an association. There are many other explanations for why that might predict future psychiatric symptoms. But it's certainly consistent with the idea that this is a risk factor. And if that's a risk factor we can do something about. I mean, we can't do much about your genetic predisposition to psychiatric disorder. It's much harder to stop warfare, right, at a societal level and prevent trauma. Other risk factors for psychiatric disorders include recent stress, but stress has an inflammation component, and we think that inflammation component is part of what's determining whether you go on to get PTSD or not, or you go on to get depressed or not. And that's something that we can do something about ahead of time. We can enhance immunoregulation and decrease the amount of inflammation in response to a stressor. So that's the theory behind what we're doing.
And so, is this the probiotic work that you're talking about? Because I know that you mentioned when we were communicating that you're now doing probiotic work. Is that what you're talking about?
It's all related. We're reserving the work with Mycobacterium vaccae as an antigen, so injections, and that's a heat-killed preparation. So we're not actually injecting live bacteria. It's killed, and so it has all the molecules that the bacterium would normally have that can act as antigens and train the immune system, but it's not living and it's not going to proliferate. There's also value, as we mentioned, about thinking along the lines of probiotics, particularly probiotics that can induce immunoregulation. And we have submitted grant proposals to look at specific probiotics in terms of their ability to either protect against psychiatric disorders or treat symptoms of psychiatric disorders. And so we think, what's not known about the ability of probiotics to enhance immunoregulation is which species are most effective, what strain of those species is most effective, how much of it do you need to consume, how often do you need to consume it, are the immunoregulation effects dependent on some other factor? And so there's a critical thing to understand about probiotics. There's a very discrete barrier between the lumen of your gut, or the center where most of the bacteria are living and eating and reproducing, and then there's a mucus layer that lines the gut, and this mucus is a fairly effective barrier to a lot of species, but some species can colonize that mucosal layer, and they can live and reproduce in the mucosal layer. And so those are the colonizing bacteria. And they're more likely to be effective at immunoregulation because somehow bacteria have to communicate with the host immune system. The gut has evolved ways of enhancing that communication. And so there's specialized cells in the epithelial lining the gut that have extensions out into the gut lumen. They can essentially sample the contents of the gut lumen and then present those antigens to the immune system on the other side. And so even dead bacteria could communicate with the host immune system through that mechanism. But the question is, are there specific types of bacteria that are more effective at communicating with the immune system through those mechanisms? H. pylori is interesting because it actually can effectively burrow through the mucus layer, and it attaches to the epithelial cells, to your cells. It attaches to those cells, and there are some strains of Helicobacter pylori that actually inject bacterial components into your cells. That's a very direct way of communicating with the host cells, attaching to those cells and then injecting material. It's also known that our immune system is monitoring those Helicobacter. So if you look at the lining of the gut, immune cells aggregate around those Helicobacter implantation sites, and presumably there's a communication or dialogue going on between Helicobacter and our immune system. They're talking to each other. That's why they're aggregating there. And it may be that the immune system is in part keeping Helicobacter from proliferating or doing other things that it shouldn't be doing to compromise our host. But we simply don't understand those questions at that kind of fine-grained detail. But you can see how the probiotics even could affect the host immune system. Those are just a few examples.
I have a question about probiotics, because my understanding is that most probiotics that we take, they don't colonize in the human gut. That's why you have to keep taking them. And so you mentioned, potentially the more effective ones would be able to live on the mucosal layer of the gut. Are there even probiotics that do that?
Yeah, certainly. I mean, there are probiotics on the market. You could go to Walgreens and buy some probiotics that are known to colonize the mucosal layer. Um, they're known to be immunoregulatory. So those are available. They're available commercially. Um, but the evidence that they have the types of health benefits that we're looking for in terms of being anti-inflammatory or immunoregulatory, we still need to develop that evidence base before we could, recommend, okay, you should be taking this probiotic, and in particular this strain of probiotic, um, if you have PTSD, for example, or if you have depression. I can envision a time when we could say that. We could say, okay, if you have PTSD, you would benefit from this particular probiotic, or a combination of probiotics. We can't do that yet because we just don't have the evidence, but certainly envision a time when that's possible. And I think even probiotics that don't colonize the mucus layer have the potential for immunoregulation because of these cells that line the gut that can actually take those bacteria into the cell, translocate it to the other side of the cell, and then present it to immune cells. There's a lot of communication, and um, for that mechanism, cells may not even need to be alive. They could be dead cells.
Mm-hmm.
And so if that's true, then there really is a constant dialogue between what we're eating and even if it doesn't colonize the gut, and what our immune system is doing.
Wow. Yeah. Probiotics as a whole, it's almost overwhelming because, like you said, it is strain
That's
specific. And then good
absolutely
luck.
overwhelming.
Yeah. And then
Uh,
good luck finding that strain. And, it
but
does, it's like the Wild West a little bit.
it is a bit like the Wild West, and I do sympathize with consumers because I've walked down the probiotic aisle at the grocery store, and oh my goodness, it's not just that you have lots of different probiotics and combination of probiotics, but different companies have different strains, and it's clear from the work of John Cryan and Ted Dinan, who are at Cork in Ireland. They have very good evidence that it's not just the species of probiotic that's important, but the strain of the species that can have health benefits in terms of emotional behavior. And I wouldn't be surprised if it's not just the species and the strain, but how that bacterium is grown. In mycobacteria, for example, you get a completely different phenotype of bacteria, a rough or smooth variant, depending on how you grow the bacterium. And so how you grow the bacterium has an impact on what kinds of molecules it's making and what's the proportion of different types of molecules. And so that's a level of resolution we simply haven't approached yet, but that's probably the level of resolution we'll need to optimize use of probiotics for health benefits.
Yeah. Wow. And one final question, just for clarification. When you say strain, like for example, Lactobacillus acidophilus, are you talking as specifically as Lactobacillus acidophilus Q135 is one strain, and then
Yeah,
Lactobacillus?
it's the Q125 I'm talking about. So Lactobacillus acidophilus is the species, the genus and the species, and then there's a little number or letter or a combination of numbers and letters afterwards that identifies the strain. And so the strains differ in terms of their genomes. They have different genes. They produce different molecules. Presumably have variable metabolism. How variable, we don't know. That's an empirical question. We'd have to test that. But those things seem to matter. The strain matters. So it's not just Lactobacillus acidophilus. It's Lactobacillus acidophilus something something something.
Yeah.
And the strains tend to be proprietary. So one company will sell a particular strain, and a different company will sell a different strain.
Yeah. Well, one final question, and then I'm going to let you go. So going back to that 10% R cells, 90% symbionts, is that 90% living in our gut, or is that all over our bodies and in our guts and other places?
That's a great question. It's not, the vast majority is in our gut, but the microbiota as a whole includes all of our tissues. And so our skin has a microbiota. The lungs have a microbiota. In females, the vagina has a microbiota. In males, the penis has a microbiota, although I don't think we know what that is yet. But all these mucus layers, all the external coverings, all of these tissues have their own complement of bacteria. And in the skin, different parts of the body have different communities of bacteria. That's clear now. And what is the contribution of our skin microbiota to our health? Obviously, it has implications for skin health, but it could have broader implications as well.
Wow. Yeah. When you think about it that way in probiotics, it exponentially expands just the overwhelmingness of it.
I think that's why there's so much enthusiasm about the microbiota at the moment. And it's important to emphasize that field is moving extraordinarily fast. There's a tremendous amount of excitement. The techniques for analyzing microbiota data are evolving all the time. They're developing. The methods are becoming more and more sophisticated. The sequencing methods are becoming more efficient, less expensive. More and more people are getting involved. And so I think, although it's a bit overwhelming because of the scale, because of the fact that this is essentially an ecology question, a community level question, it's complicated. We can't ignore the fact that it's complicated, it's intricate, but it has vast potential. That's what's so exciting about it. And I think realizing that potential is all about understanding the specific mechanisms through which these microorganisms communicate with the host, not just with the host immune system, but with other organs like the brain. How do the microbiota communicate with the brain? It's clear that they do, but we're still understanding those mechanisms that are involved.
Well, I want to thank you so much and for talking with me today. And I look forward to seeing your work with the probiotics as you move forward. What kind of timeline are you looking at?
Well, with the injectable Mycobacterium vacui, we've done a lot of work that we should be able to publish that soon. With the probiotics, we're really at this stage of designing clinical trials and getting regulatory approval, that kind of thing. And that just takes time. It's a bit unpredictable, so we don't know.
And so with the Mycobacterium vacui work that you've done, that you're ready to publish, that's been its association with psychological, with basically the brain.
Yeah, we're really interested in how enhancing immunoregulation by using these immunoregulatory bacteria can affect emotional behavior with implications for psychiatric disorders like anxiety and affective disorders. So that's our focus.
Got it. And when's that coming down the pipeline? When will that be out?
Hopefully you'll see it within months.
Okay. Yeah. All right. Well, thank you so much, Professor Lowry. I really appreciate it.
It's a real pleasure.
All right. Have a great day.
Okay, thanks.
Bye.
Bye-bye.